And I'm going to be talking about my project, or kind of bits of my projects, which is phages and their ecological strategies to advance phage therapy. So obviously we're all kind of here for the same reason, but one of the main reasons is obviously we're looking for alternative antimicrobials, which is bacteriophages. But one of the issues when we come to develop a cocktail is that every time we go through this kind of long process of host range testing and testing the efficacy of the phage, and this can be quite time consuming. So to avoid this bottleneck, obviously the best way to do this is understanding of phage biology and understanding the phage traits in terms of what make them successful at doing their job. And this will hopefully help us streamline the selection of clinically favorable phage. However, there is a bit of an issue, and this is that many genes are uncharacterized in terms of what they actually do. So these are two of my, or one of my hosts and my phage that I've been working with. And if we just look at the number of hypothetical proteins, for example, in the E. coli genome, there's only 18%, whereas in my phage, there is 44%. So there's obviously a big gap of knowledge in terms of we don't know what these exactly do. And my project is hopefully, or parts of my project are trying to sort of understand these genes and maybe try and characterize exactly what they're doing. So to start with, I'm going to characterize the phages in a therapeutically successful UTI cocktail. This was a cocktail developed by Mel Haynes in our lab. And then moving forward, I wanted to use transcriptional profiling to try and identify these phages into three groups: early, middle, and late. And the reason we want to do this is because we don't really know whether the proportion of the early, middle, and late genes has an impact on the therapeutic potential. So we want to try and really understand why phage do what they do. And then finally, we want to use this knowledge. I won't go into this too much today because I only have a short talk, but to develop an ecological framework to try and group these phages together. So one thing I'm interested in is the lytic cycle, and I'm interested in key time points during this lytic cycle. So I've been doing a lot of absorption assays and one-step growth curves, especially one-steps. I feel I could do them blindfolded, honestly. But something I'm interested in is obviously the initial absorption and injection of the DNA. And we're interested in this early time of the lytic cycle because we feel that obviously a number of genes here will be linked to sort of interaction with host systems. Towards the end of the latent period with assembly and lysis, later in this sort of phase, there will be genes linked to assembly proteins and lysis proteins. And then also during the eclipse period, when there's no virions detected, there should be genes linked to sort of the viral synthesis or modulation of the host. So I've been performing absorption assays. Just for this talk today, I'm only going to talk about two phage. One of these phage, called JK08 there, belongs within this UTI cocktail. And then the other one, called phage 113, was previously actually in this cocktail, but then was removed because it didn't perform as well as a few of the others in there. In this cocktail, there's three phages, and I'd say that one was removed. But the main thing I just want to highlight on this phage that, this page is that I did some absorption assays, and after 10 minutes, both 90% of the phage had absorbed in both of them, or over 90%. However, initially after 1 minute, in 113, this was much, much lower. So obviously you can already see that they have slightly different strategies they go about doing things. Moving forward, I would then do one-step growth curves. And as I mentioned, I've done hundreds of these, or what feels like. And I was doing this at both 0.1 and MOI5. So because I'm doing transcriptional profiling, I come to do RNA extractions. And I wanted to ensure that I had a significant number of cells infected with phage. So I do this at a high MOI. I also did it at MOI 0.1 because kind of 0.01 and 0.1 are like people do standardly. And I wanted to see if there was any difference, which I didn't find any difference particularly in when I was doing these. And then I also wanted to note on this is that these two phage both had slightly different latent periods. So again, showing slightly different strategies, let's call it. So following this, I went on, I extracted my RNA at different time points, and then I sequenced them. And then I got these lovely heat maps, and I then clustered the genes into 3 transcriptional phases, which is early, middle, and late, based on their time. And quite simply, like the red is high expression and the blue is low expression. And I've tried to group them, so obviously it's kind of hopefully obvious to see. So I then did this, and then I wanted to count the number of genes that were maximally expressed in each phase. So I had 98 maximally expressed early, 120 maximally expressed during the middle, and then 61 maximally expressed during the late. And then I basically repeated this and did the same thing for my other phage, which was 113. Although this one was a little bit different from the other, but I basically repeated the same thing, clustered them into 3 groups, and then had a look at the number of genes again. In the case of phage 113, I had 281 genes, which 6 were maximally expressed early, 195 middle, and then 80 of those were late. So obviously these two appeared to have very different ways they were going about doing it during the infection cycle and had different strategies. So when we compare both of them, like when I looked at them initially, like JK08 looked like it was kind of even across the board throughout, whereas 113 seemed more of a specialist, is the way I determine it. And this might be one reason in which 113 was previously in the cocktail, but then removed. So maybe a more general or jack of all trades phage was more suitable for this cocktail that we've developed. So yeah, it's both of those. So like, I only have a short talk here, but the main kind of gist of my project, which I haven't gone into too much, is that we're trying to categorize these phage into 3 groups using this plant framework. So this was a framework that was previously used to classify plants into these 3 groups. And essentially, we want to do the same thing, but within phage. So hopefully we can end up with something like this, in which we have our early, middle, and late, and we have phage on here. And hopefully this, by understanding and grouping the genes and hopefully being able to categorize them, we can hopefully identify what traits are best for cocktail development and those that are most therapeutically efficient or successful. I have a few other things that I need to do, so I've got like a lot of work, too much sometimes. But I need to repeat some experiments, and I have other phage that I want to get the transcriptomes of, so I need to also do those as well. I need to analyze host response, something also we're interested in. And then we've got our other available transcriptomic data we want to use as well that's out there, so that we can also add this to this framework and hopefully bolster it more to identify some key predictions possibly. That's me done. Thank you for listening. Thank you to my supervisors, everyone in Lab 230 who's always very helpful and helps me deal with the stress, and MIBTP, which is the doctoral training program that funded my project as well. Thanks.